Naive T cell activation stimulates memory cells and helper T cell production. The actual acquired immune response originates with the innate immune response and the entry of antigen into the body. At their site of entry or within the lymphatic system, the antigens are phagocytes by the APCs, such as dendritic cell and macrophages. As the cells migrate to the lymph node or other secondary lymphoid tissue, the antigens are broken down and peptide fragments are displayed on the surface as MHC2 or peptide complexes. When the APCs enter the lymphoid tissue, they mingle among the myriad group of naive T cells, searching for the cluster having the surface receptors that recognize the MHC2 or peptide. This process requires considerable time and energy because only one cluster of T lymphocytes may have matching T cells or CD4 receptors. It is important to remember that recognition is between the MHC2 proteins on the APC surface and receptors on the T cell surface. It is as if the T lymphocyte must first ensure it and the APC are from the same body, means same MHC proteins, before it will respond. The CD4 co-receptor enhance binding to the naive T cell to the APC because the CD4 protein recognize the peptide associated with the class 2 MHC protein. Bound to the T cell, the APC secretes specific cytokines such as interleukin-1, which binds with the naive T cell and stimulates T cell activation. Interleukin-1 causes the naive T cell to secrete other cytokines, including interleukin-2, which stimulate cell division of that T cell and others activated by the APCs. The result is production of a clone antigen-specific activated T cells. Some of these mature into memory T cells and await a future encounter with the same epitope. The majority of activated T cells mature into one of two types of effector T cells, depending on the influence of specific cytokines. Some mature into helper T2 cells. Helper T2 cells help the activation of humoral immunity. Other immature T cells mature into helper T1 cells. These cells recognize and bind to infected APCs, displaying the appropriate MHC1 or peptide. Cytotoxic T cells recognize MHC1 peptide complexes. Activated cytotoxic T cells seek out and kill virus-infected and abnormal cells. Because body cells also can become infected with viruses, the immune system has evolved a way to attack virus-infected cells. During the infection, the host cells manage to degrade some of the viral antigens into small peptides. These are attached onto MHC1 protein and transported to the cell surface, where they are displayed like red flags to denote an infected cell. Activation of naive CD8 T cells occur through interaction with dendritic cells or macrophages displaying a MHC1. Activation also requires interleukin-2 stimulation. Both events activate CD8 T cell to divide into mature cytotoxic T cells, able to recognize the antigen being red flagged on infected body cells. The cytotoxic T cell leave the lymphoid tissue and enter the lymph and blood vessels. They circulate until they come upon their target cells, the infected cell displaying MHC1 on their surface. The CTL bind to the MHC1 or peptide on the virus-infected cell surface and release a number of active substances. Toxic protein called perforin insert into the membrane of the infected cell, forming cylindrical pores in the membrane. This lethal hit releases ions, fluids, and cell structures. In addition, the CTL releases granzymes that enter the target cell and trigger apoptosis. Cell death not only deprives the viral pathogen of a place to survive and replicate, but through cell lysis also exposes the pathogen to antibodies in the extracellular fluid. Cytotoxic T cells also are active against tumor cells because these cells often display distinctive molecules on their surface. The molecules are not present in other body cells, so they are viewed as foreign antigen peptides. Harbored within MHC1 proteins at the cell surface, the antigen peptides react with receptors on CTL and the tumor cells are subsequently killed through apoptosis. However, some tumors reduce the level of MHC1 proteins at the cell surface, which impedes the ability of cytotoxic T cell to find the abnormal cells. Thus, tumor cells can escape immunologic surveillance and survive. helper T2 cells initiate cellular response to humoral immunity, and the active helper T2 cells co-stimulate B cell activation.